We're going to be talking about eukaryotes' gene expression. So let's get right into it. For eukaryotes, their system is not coupled together like prokaryotes are. It is decoupled because they have the different organelles that separate the DNA in the nucleus from the ribosomes that are out in the cytoplasm. So their process is a lot more complicated. So they have a lot more opportunities to regulate the process, which allows for a lot more regulation and editing so that there is not as many errors. So if we were to look at the whole process all together, we can see all the different control points here. Oops. Here, and here. So you can see here in the nucleus, here is the chromatin. Remember, chromatin is a mixture of DNA and histone proteins that is loose and not compacted. This is the way that DNA is when it is not going through cell division. Here is a simplified version of the DNA, and we can see that a piece of that DNA has a gene on it. Okay? And that gene is available for a transcription. After it's transcribed, you're going to have the primary transcript of mRNA. Notice there are areas of peach and red. That is showing you the introns, the non-coding pieces of DNA, and the exons, the coding pieces of DNA. After those introns are cut out through RNA processing, you see the exons have all been coupled together, and there has been added a cap and a poly A tail. This is to protect it as it goes out of the nucleus into the cytoplasm. Here you can see the mRNA is now going to do translation, but if this mRNA has any mutations, there are ways for the system to degrade it, to get rid of it if there's any issues with that mRNA. So we'll talk about more how that happens. If the mRNA is okay, it can go through translation and make a polypeptide chain and fold into an active protein. But if that protein is no longer necessary or has a mutation, there are also ways for the system to degrade that protein and get rid of it. So you guys will be doing a PhET simulation, and you'll be able to see this all in action, these different checkpoints where they're degrading the mRNA or degrading the protein when it's not being used. But let's get into the very beginning of how this DNA is actually going to be expressed in the first place. So first, let's talk about DNA access. Remember that all of your cells have all of your DNA in it. They have every single gene that you are supposed to express, each and every single one of your body cells, your somatic cells. But only some of those cells are expressing the specific gene that they need to. They are differentiated. Once your cells become differentiated, they have to only follow specific genes. They're only expressing specific genes for their whole entire life. This is how a eukaryotic cell or a multicellular organism controls the cells to do just what they need to do in that portion of their body. So each of your cells have become differentiated, and each of your cells are only following specific genes to do their specific function, and they're ignoring the rest. But how can they do that? How are those off? How are those genes off? So let's talk about that a little bit more. When you look at your DNA that's loose as chromatin, it is actually wrapped around these little purple circles here called histone proteins. And there's also these little histone tails that come off of those histone proteins. And when methyls, CH3, attach to those, here you can see these green dots are acetyls, but if those were methyls, it would make the DNA be more tightly packed like this. Okay, so they'd be unacetyled and they would have methyls. When DNA has acetyls, or the histone proteins have the acetyls on them, the DNA becomes more loose. And you can see here there's this looseness and there's these areas that are exposed of the DNA that can be more easily transcribed. So when your gene is off, it's going to be looked more tightly packaged, and the methyls will be added onto the histone tails. Where when acetyls are added, they are going to be more loose and more easily expressed. So in those cells that have the gene off, it looks like this. And they are not, the RNA polymerase cannot access the DNA to transcribe it. And in your cells that have the gene on, it's going to look, your chromatin's going to look more like this, and the genes are able to be expressed. And so this is a great way for the cells, the organism cells, to control what genes or what cells do, okay, what their main function is going to be. So this also, though, is how epigenetics can come into play. So you can see here, cool fact, histone acetylation patterns seem to be heritable. So we can also inherit some of these loose DNA situations, and that's how some of these genetic disorders that can be passed on later on in life, because you have this acetylhistone loose in a certain area of your DNA that most people would have it closed. So this would be epigenetics. Also, things in your environments could increase the amount of CH3 methyls to make it tighter again, or something in your environment could increase the amount of acetyls. So that's what epigenetics is. When your environment shifts or changes, your gene expression. And so that's what we call, so it's not your DNA actually changing, it is the acetylation that's changing, and that can affect what genes you actually express, and that is epigenetics, when your environment is controlling what genes you express. But just remember, your DNA doesn't change, it's not mutated, it's actually what's attached to the proteins around your DNA, the histone proteins. So this is one way, or two kind of ways, that your cells can differentiate as well as affect gene expression. This also is pretty interesting. If you look inside the way DNA is wrapped around each other, they face inward. Some of the DNA will loop inward, and we call these transcription factories. And these are usually places where there are different genes that all have same roles, so they all usually need to be transcribed at the same time, but they're not near each other on the DNA, but they group together like this into areas on the chromosome or near each other in the chromatin, so that they can all be expressed at the same time because they're not in order on the DNA. So we call those transcription factories. All right, another control point is in pre-transcription. So this is how eukaryotes actually transcribe their genes, as opposed to operons, which is how prokaryotes do it. So all the actors need to be present for the play to begin. So let's talk about those actors. So we still have the promoter region, which is where the RNA polymerase is going to attach so it can transcribe this gene. And you can see this is the termination point here at the end of this gene. There are still an area up ahead, which we call the enhancers. So the enhancers are going to be in charge of getting this process started. In an operon, we call that a regulatory gene, right? But in a eukaryote, we're calling it an enhancer, okay? So these enhancers, in order for them to begin, there needs to be something that attaches to them. And we'll talk about that more in a second. So here is a picture of all of the actors in play. So here is the first image. Here are these enhancers that are upstream in front of the promoter. The promoter is usually like a sequence like TATA, and that RNA polymerase can know or recognize and attach to it so that it can start expressing this gene. These are called activators. So activators is different than the activators that we talked about in prokaryotes, where the activator actually increases the transcription of the prokaryote operon. These activators are necessary for transcription to happen at all in a eukaryote. So you'll notice these these activators attach here to the enhancers. Once that happens, transcription is initiated. So that would be the beginning of a eukaryote's transcription or gene expression. Then this DNA bending protein is going to come here and bend the enhancers and the activators so they're closer to these other transcription factors that are necessary for them to attach to. So this DNA bending protein bends that line of DNA so that they can loop and attach to these mediator proteins and these transcription factors. So all of these I would consider transcription factors, and they are going to attach to the activators, and it's going to create this structure right here. This transcription initiation complex is what we would call that. Once they are all attached to each other, RNA polymerase can then attach to the promoter, and it will move forward down the gene, and it will transcribe it, and RNA synthesis will occur. So remember, enhancers, upstream from the promoter. Activators are needed to attach to them, and then they will then the DNA bending protein will arrive and bend the DNA so it can attach to the transcription factors. And then once they are all attached to each other at the transcription initiation complex, it will initiate, it will start transcribing. So if these activators aren't present, that will control if transcription can occur or not. So it's another way for eukaryotes to control the process. This is actually how differentiation occurs. So if you notice, this is a liver cell, and this is a lens cell, like in your eye, a lens. So each of these have the same setup. They have enhancers upstream of their promoter, and then they have the gene. So in the liver cell, they want to make albumin, which is a protein, and in the lens cell, they want to make crystalline gene. Now here in the liver cell, you'll notice that these activators match the enhancers, the colors at least, of these enhancers that are on the DNA or are on the DNA upstream of making albumin. So they match and they attach, and then they are able to create this transcription initiation complex, and transcription of albumin occurs, and this gene is expressed. This gene is also present in the cell, the crystalline gene, because your DNA, all your DNA, every gene you have is in every single one of your cells. But there are no activators present, so they are not going to be able to turn this gene on. So either this gene is not able to be reached because of it being methylized, and so it's tightly packed, or just the correct activators won't be present in the cell ever, so this gene won't get turned on. So in the opposite scenario, here is the lens cell nucleus, and you can see the opposites happening. The activators actually match the enhancers in order to, down here, the enhancers to make crystalline, and so this activator is attached to the enhancers on the DNA. It creates the transcription complex, and the RNA polymerase is then able to transcribe crystalline. But notice this activators that are needed to make albumin are not present, so this gene will not be expressed. So this is an extremely important reason for how, or way that cells are able to differentiate and only make the lens cell do what it needs to do, your liver cell do what it needs to do, and not every function in your body in one cell. So this is how another way that cells can regulate and continue to differentiate from each other. Here's post-transcription. So post-transcription we've already talked about a little bit. So remember that after the mRNA is transcribed, it also transcribed the non-coding portions of DNA, the introns right here. So it's going to cut or splice out those introns, and then it's going to glue together the exons. Also notice after it glues together the exons, it adds the cap and the poly A tail, and this is going to ensure that the mRNA remains functional as it's transported through the nucleus and the nuclear envelope pores to the cytoplasm for translation. It just protects the mRNA. So here is the alternative splicing, which we've also mentioned before in a previous unit. But you can see the exons, they've given them numbers, and the light blue are the non-coding portions of DNA. After they've been transcribed, because we don't want to damage the DNA, we can cut up and do what we want with the mRNA because it can just be made again. So the peach areas have been cut out, the introns, and it has other enzymes spliced together in the order that they need to, the exons. So notice number 4 has been left out of this mRNA strand, and number 3 has been left out of this one. Because each of these are different genes. As you saw in the lac operon, there's like 3 different genes right next to each other. So they're going to make 3 different proteins. Sometimes these proteins will then combine together and make a quaternary structure with multiple proteins. Sometimes they'll each have a different function individually. And sometimes you don't need one of them. So this can create variation within how the proteins are made, and it can be faster and more efficient, right? You can get more proteins and different ones from the same gene expression without having to do it again to get the second option that you would like. So anywhere from 75 to 100% of human genes have multiple exon probably undergoing alternative splicing. So shown here is the troptin T gene, which produces 2 different mRNA sequences to produce 2 different gene products. All right, so another area that there can be regulation on how genes are expressed is pre-translation. So this is before the proteins are made. So this is a new topic that we haven't talked about before, but those introns that we talked about, those non-coding pieces of RNA, they actually have a function. So there are more RNA types than we had realized. So if you look here at some examples, here are the ones we talk about for coding RNAs, like messenger RNA, rRNA, tRNA, transfer RNA. These are all ones that help with synthesis. But there are also RNAs that are regulatory RNAs that are going to help control and make sure everything's going right in the cell for gene expression. So RNAi is the one we're going to look at. RNAi stands for interference RNA. It's basically like a cop that's going to go around and it's going to look for any mRNA in the cytoplasm. So it's in the cytoplasm, it's not in the nucleus. Any mRNA that's made it out after the processing of cutting out the introns and putting together the exons, it's going to look for any mRNA that has something wrong with it, it has maybe a mutation, something's not right, and it doesn't want it to be made into a protein. So the RNAi is going to degrade mRNA that it sees, or it might just hang on to that mRNA so it blocks translation from occurring at the ribosome so that protein will never be made. So there are also microRNAs or miRNA, which is what tags it so it knows what it's looking for. You're going to watch a video to understand this a little bit better. After you watch this video in Edpuzzle, you're going to be watching that one and answering some questions. So you'll learn more about that there. All right, and our last area for regulation is post-translation. So after the protein's made and you think, oh no, this bad protein is already in my system, what are we going to do? There's also a way to get rid of that one. So if that gene actually expressed that protein it's wrong, it's misshaped, it's not folded right, it's going to have a bad effect, it's not going to do its function. There is an organelle called a proteasome, which can be tagged, or which is going to go around and degrade those proteins that we don't want into different peptide fragments. So the way this works is there is this molecule, a protein, everything's a protein, right, called ubiquitin. And ubiquitin is going to attach itself to any protein. It's like a tag. And it's going to say, hey, proteasome, come over here. You don't want this one anymore. And it's going to proteasome will come and it will break down that protein and degrade it into protein or peptide fragments. So this organelle is very abundant in eukaryotic cells because we need these regulations. Our system is constantly making proteins, and we need to be able to get rid of these proteins that are not working, or maybe they're no longer functioning, maybe just that they no longer have a purpose within that cell. So there, as you can see in this picture, these are all a bunch of proteasomes in a cell. So they are abundant and they're very much important to our system. So there you have it. That is how eukaryotes do their gene expression. And please make sure that you've answered all the questions in the Edpuzzle, and I hope that was helpful.